Please take a moment to download the high-resolution image of this from the Glossary of Leaf Morphology Terms on Wikipedia.
Hey everyone, so welcome to the Leaves Lab. In today's lab, I do expect y'all to be pausing the video a lot. We need to do a lot of leaf drawing. Now, of course, that requires a still image of the leaf. So at any point in this lab, please make sure to be pausing it as you need in order to copy down leaf patterns. Because the goal of today is to understand how variable leaves are, and you will recognize this poster from the Glossary of Plant Morphology Terminology. It was also the panel just ahead of the start of my discussion here. You have access to it from Wikipedia, you have access to it in the lab. I do not expect y'all to have these terms memorized, but I do expect you to be able to identify the different patterns. Because leaves are highly variable photosynthetic organs. And we're going to try to put a little bit of vocabulary on the basic anatomy of those leaves underneath our belt today. So that's going to require us to take a look at some living plants. So let's talk about the basic structure of a plant leaf down over this way. So, our first order of business is to discuss what is a leaf. That means that we have to go over the basic terminology of a leaf. You have the flat edge of the leaf, the blade, the lamina, the sheet. This is where your photosynthesis is happening. You have the stem that connects our leaf to the stem of the plant itself. The stem of a leaf gets a different name. It's the petiole. And then on every one of our simple leaves, you will get a stipule, an outgrowth at the base of the leaf. That's the key thing in order to determine whether this is a simple or a compound leaf. Because a simple leaf will have a lamina, petiole, stipule, versus a compound leaf, like that which we see here, where you do not see a stipule at the base of every leaf. You instead see the stipule on the outgrowth of the base of this leaf. So this whole structure here is actually a compound leaf. This structure here is a simple leaf. So hopefully that helps illustrate the differences between the two. Simple have got stipule at its base. Compound leaves have many leaflets with the stipule at the base of those leaflets. And so that's the basic terminology we're going for when we're trying to understand the variation in terms of what's the organ that grows out, right? To the same extent, so that way y'all can kind of put it into human terms, a hand is a combined organ of many fingers, right? In a simple leaf, this would be the equivalent of the entire hand. It's melded into one. In a compound leaf, each of our leaflets branch out. Make sense? Cool. The next major delineation in terms of our leaf structure has to do with the patterns of the veins. And we will be staring at vein patterns a lot today. There are two basic groups of vein patterns. The netted veins, meaning those that kind of look like a net. They go everywhere. And then, come with me, the parallel veins. Veins that run parallel to one another, like in this palm tree here, the Kentia palm. The parallel veins belong to the monocots, the group of flowering plants. Yes, the palms do have flowers. The group of flowering plants that are that only have one cotyledon, oftentimes things like our grasses, and are distinguished by having their parallel veins. In contrast, our netted vein plants are our eudicots. They have veins that sprawl across and fall into two different categories. The first is that of our begonia here. These all come to a single point, like on your wrist. Hence, we call this netted pattern of this begonia palmate. Contrast that with the netted pattern that we see in our money tree here. One main axis where they all connect. If they're all connecting, each of those branches are called a pinna. So this pattern is pinnate. Contrast that against this pattern that is palmate. What do y'all think our pattern is for our beautyberry, the calicarpa? What do you think, Mr. Odie? That nose definitely says that this is another pinnate leaf. So that gives us our basic idea of our major structural breakdowns. You've got parallel versus netted. You've got pinnate and palmate within the netted, parallel doing its own thing. And next up, we'll explore a wide diversity of the terminology for our little leaf variants. So I'll see y'all in a second. Okay, so here I am with a couple of different leaf samples laid out where we're gonna kind of take an opportunity to stare at some of the vocabulary, stare at some of the glossary of plant morphology terms, and see what we can identify. So first up, let's take a close look at this here beech tree sample, Fagus grandiflora. These are samples actually generated during my local flora class in the summertime. And I think it's a great illustration of the difference between simple and compound. Now you will notice that this leaf is definitely pinnate, right? It's got one main axis with all the veins coming off of the sides. But is it a simple leaf or is it a compound leaf? Well, in order to identify that, we need to see where the blade is, we need to see where the petiole is, and then we need to see if there is a stipule at the base of the petiole. And sure enough, to me, that looks like a stipule. Let's take a look at another leaf. Do I have a stipule, an outgrowth at the base of the petiole? Yep. 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 Each one of our leaf blades on this Fagus grandiflora has a stipule at the base. You even see that one of those stipules has instead grown out to be one of the beech nuts, the reproductive structures. And they can do that. Now contrast Fagus grandiflora with our horse chestnut. Here you have a leaf. It's got leaflets, or blades that are separated rather. You see a pinnate structure to this as well. So you'll notice that it's got one main axis, all the veins coming off of the sides. But where is our stipule? We don't see a stipule present here where all the leaflets are connecting. Instead, the stipule would have been present as an outgrowth here at the branch point. So this is no longer attached to the tree. It's dried off and fallen off, but the lack of a stipule present around our connection here suggests that this is a compound pinnate leaf versus a simple pinnate leaf. Now additionally, just to layer some more vocabulary on here, each leaflet of our horse chestnut is pinnate, but look at the overall layout of the leaflets. They sure do look like the palm of my hand, right? This means that this is actually a compound palmate leaf with pinnate leaflets. Now that ought to give you a headache, right? There's a lot of vocabulary to go in each of these in terms of leaf morphology. So let's take a quick tour of some of the other diverse terms, and I'd encourage you all to pause the video as you go before I announce it to see if you can take a guess based on that poster of the glossary of leaf morphology terms. So have that open in another tab if you will. And let's see if we can guess or agree upon what each of these shapes or general structures are. So let's start with something that's one of my favorites in the area. This bedstraw, Gallium species. Know the genus, don't know the species. You'll notice that each of these leaflets are coming off of the center of the stem. When you have many leaflets coming off the center of the stem, we call that a, pause the video, or not, that's a whorled leaf. Whorled around the center, looks just like, if you want to pan up this way, the bottom right corner there. whorled leaves all around the center, coming from a single point. Now next up, let's see if we can use a single vocab term for each of these. You'll notice this cattail leaf, Typha angustifolia, the narrow-leafed cattail. It has veins that are parallel. So ignore the pinnate palmate. It is one of our monocots. Very cool. Now next up we have the common milkweed, another plant that you'll see here in the area. Do you think that this is pinnate or palmate? Well, I'm seeing that central pinna, right? One main axis, one main vein. Compare that again to this star flower, Trientalis borealis. Its name's changed a couple times, even in my time here at West Shore. You'll see many different leaflets coming from a central axis. So each leaflet is pinnate, and each whole leaf together is a compound leaf, because there are not stipules at the base of each leaflet. We've got a compound palmate leaf with pinnate leaflets. Now for something that you might recognize when you look up, we have our swamp white oak, an oak tree. Looking at this leaf shape, are we looking at something that's pinnate or palmate? Again, another pinnate structure. But you'll notice that the margin is very unique here. We are looking at something that is lobed, very, very large teeth on oak leaves. That's what defines them in a big way. Now last up, we have juglans, the black walnut. It has many leaves along an axis. And let's zoom that camera in to see do we see a stipule at the base of each of those leaves? I see no outgrowths at the base of each of these leaves. This makes me assume that this is actually a compound leaf. One main axis, it's a pinnate compound. So contrast our pinnate compound and our palmate compound. Leaflets emerging from a single point, leaflets emerging from a single axis. So next up, we'll take a little bit of a tour of some of the other plants that have been preserved in the classroom. You'll notice this board actually comes from a little bit high up, so next time that you're in the classroom, I'd encourage y'all, it'll be hanging up there next time you're here. I will just have a slow meander through all of these and ask if you can identify any of the shapes. So pause the video as necessary to look at our Norway maples, or our tulip leaves, both of those looking fairly palmate to me. Perhaps our black cherries, elm, some of our more pinnate leaves, a palmate Japanese maple, the black locust with a pinnate structure, our aspens, the cottonwoods, the poplars, the same genus of plants that go by 3 different common names, a botanist's favorite. Again, some more pinnate beech leaves. And then here in our sassafras, we've got some variation in leaf structure. Sometimes 3-toothed, sometimes mitten-shaped, sometimes without a single lobe at all. In terms of the most unique structure here on our leaves, we have our ginkgo plant, fan-shaped. These have parallel veins that run together down to the very middle. This is actually a cone-bearing plant. Ginkgos are super cool, and we will spend a lot of time talking about Ginkgo biloba, the last remaining relative of this entire branch of trees. So hopefully you've had an opportunity to pause here. We'll again, in a moment, take a tour of the greenhouse and see if we can tag on a couple of more leaf shape terms to some of the things that you'll be seeing in the classroom. All right, hey everyone. So we're here in the greenhouse. We're going to be talking about a rose first. We're on this mission to see if we can identify simple versus compound again. So a rose has some leaves, but are these leaflets or are each of them leaves? Do we see a stipule at the base anywhere? Or is this truly the petiole at the base of it? Well, if you look down here, you will finally see an outgrowth at the base of the stem. So the stipule is combining this, and this as our compound leaves. So roses are highly compound, so roses are highly compound because you can identify where that stipule is at the base of the petiole for many different leaflets. We can keep walking around the greenhouse. We see some of our favorite little ferns. These all having pinnate leaf arrangement. Now some of them will be alternating with one, then two, then one, then two, then one, then two. And some of them will be a little bit more opposite, where they have leaves on each axis. This one is not opposite, but again, a little bit alternating. We can look at our banana tree and see all of the veins here coming off of one main axis, even though this banana is a monocot, which typically have our parallel leaf venation. We see a little bit of variation within one of the relatives of the grasses. Now we can continue, see some sedges with our parallel veins here, papyrus. Our beautiful philodendron, a member of the Eraceae, another group that is a monocot but has some netted venation, quite pinnate if you ask me. And then we have probably one of our most clear stipules that you'll see in the greenhouse. So this croton has beautiful vegetation. I think so, at least. And you can see that each of these leaf blades that are pinnate have a little bump at the top of them. Those are our stipules for a croton. So petiole, blade or lamina, and stipule, defining our simple leaves. You can continue on your tour. Don't trip over our little puppy. See another parallel veined plant, a bamboo, a monocot, a cousin to corn. So you see all the parallel veins on this beauty. And then from there, we'll take our next discussion, a discussion of heteroblasty, the phenomenon that is simplified as plant puberty, where plants will have different leaf shapes when they're young versus when they're older. Pines are a great example of this. You'll note on some of our younger pine seedlings, look at the space between each of the pine needles. You can actually see some stem there. Compare our first year pine seedlings and how spacious you have, how much space you have between each of the needles, and some of our older 2 to 3 year old pine seedlings. You see how many more needles come from each emergence on the stem. As the plant gets older, it'll start to accumulate some more sugar. When it accumulates more sugar, it is changing how frequently it's building its leaves. Heteroblasty. I'll meet y'all over on the slideshow to talk about the phenomenon of heteroblasty by looking at it in some experimental setups, and then some of my own images from hiking around in the summertime to see how leaf shape changes between some seedlings and some grown-up plants. See y'all inside.
So as promised, here we are with some images of different plants in their juvenile and adult forms. We see that the leaf, both texture, size, shape, changes quite drastically between the young and old here. In conifer plants, heteroblasty is particularly pronounced, where we see drastic changes from the young and the old leaves on the same exact plant. Needles versus awls on the same plant. This is, again, carried over in a lot of our other conifers, as we discussed in the greenhouse, we see this image from the lab proper showing early growth in pine needle internodes, and then seeing how much more condensed they are, how many more bundles they're setting up, and how the needles change through life. We also see heteroblasty present in fern lineages. This is the water fern, Marsilia. When grown in low glucose concentrations, they will not set out leaves as seen, but then in high glucose concentrations, they will start to set out leaves. And of course, as discussed in the lab, we talk about a whole plant kingdom approach to how leaf shape changes throughout life. So in this image, what you're looking at are the different groups of plants, everything from some early algae to the mosses. Even mosses have some changes in leaf shape, to the fern lineages, Marsilia, to some of the monocots like maize, and then lots and lots of different eudicots. Our daisy family, our roseate family, and our mustard families. And from left to right on each of these, you can see youngest leaf to oldest leaf, like in grapes. Eucalyptuses have changes in time. Cotton has changes in time. Even Arabidopsis has change in its leaf morphology through time. So take a moment to appreciate the wide diversity, and also this is probably a good pausing moment to check out the questions that are present in the lab.
All right, so real quick, before we head into all of our microscopy on the class, I'm going to take advantage of the fact that we have this beautiful leaf model in the classroom. Now, the leaves that we were looking at from afar, the leaf shapes, are simple and compound leaves, pinnate and palmate, all the variation. It carries over to the internal structures because those leaf veins correspond to where you'll see xylem and phloem. And we see a lot of variation that mimics the metabolism of the plant, or that's necessary for different plant metabolisms. And so here we're looking at the basic anatomy of a leaf that has C4 photosynthesis. Now, y'all will remember C4 photosynthesis from our lectures earlier in the semester, where you have the light reaction occurring in a separate tissue layer from the dark reaction, where rubisco is grabbing onto CO2, where CO2 concentrations are high, closer to the spongy parenchyma, and where there's a whole bunch of light coming in our palisade parenchyma. From top to bottom, the tissues that we're seeing, the cells that we're seeing, you'll see an epidermis. It's got a cuticle on it, a waxy covering. You will see fewer stomata on the top, the guard cells, and the stomata itself, the opening, the pore. But this is all part of that single epidermis that helps prevent water loss. Now below that, you will encounter the palisade parenchyma, the fence-like parenchyma, our metabolically active, photosynthetically active, lots of the light reaction occurring in these, lots of chloroplasts. All of the ATP and NADPH generated here will be shuttled over to our bundle sheath cells, the tissue layer around the vascular column, our vascular column being made up of our pink xylem, see the bigger vessel elements, and our teal phloem. Because when you're peeling this off of the stem, where the xylem was in the middle, phloem was on the outside. When you plop that over, the xylem's on top, phloem goes on the bottom. The bundle sheath are in yellow alongside some of the supportive parenchyma. So again, fibers that are supportive. And then below this, we have our spongy parenchyma, the tissue that's very airy, that's going to enable us to do a lot of gas exchange. because you're seeing a whole bunch of stomata on the bottom of our leaves. This is where the gas exchange is happening to let that air in. Now, this basic anatomy, C4 anatomy, is going to be innovated upon in a lot of different ways in a lot of the plants that y'all are going to see under the microscope in a little bit. Make sure that you're paying attention to the bolded terminology, the different leaf structures, as we'll be taking both a look at some of the basic blueprints for leaves and then some of the adaptations that we see in different plants, like water lilies or dry adapted plants. I'll see you all at the microscope.
With our microscopy images from our first leaf samples in the lab. The lab instructs you to first grab a lilac leaf, syringa, and looking at it at 100x, we can see a lot of the different terms being discussed. Now your task here is to pause the lab and draw our cross sections of leaves and label according to our guidelines. Keep in mind, a lot of the terms sometimes will be listed here on the right, but also are listed in the lab. You will be labeling these with a ruler, making parallel lines off to the right side, so that way they all end at the same point, not like the terms are listed here. And then make sure that your label for the material is placed underneath and magnification is adjacent to it. I will highlight some of these things with my cursor. So we of course know the epidermis on the upper side and on the lower side are single cell layers that help keep all of the contents on the inside of the leaf in. We have the two different types of mesophyll, our palisade mesophyll that looks like a fence, and our spongy mesophyll on the bottom of the leaf that's much more porous. We of course see our conductive tissues, the pink xylem and the lighter green phloem beneath it, tealish in color. Of course, we have that circumstance in our leaves where the xylem is on top, phloem is on the bottom because they are an extension of the stem. We see a cuticle layer, a waxy layer that gives the top of the leaf as well as the bottom of the leaf a little bit of a shadow. We see some stomata. We'll take a closer look at those in a second. And of course, we have our air spaces in our spongy mesophyll. Here is a close-up on the vascular bundle at 400x, where you can see in greater detail the structures of the vessel cell walls, as well as the distinction in some of the phloem tissue. You can even see some of the chloroplasts that would have been present in some of the parenchyma, the spongy parenchyma, and even more of those chloroplasts in the palisade parenchyma. We have some of the parenchymal tissue that is supporting our main vascular bundle. And then here you very distinctly see the waxy outside, the cuticle on the surface of the leaf. Now scanning over on the microscope, you would have found a thinner section of the leaf where more of the stomata are contained. And here we have a pair of guard cells that are currently closed. This stomata would lead backwards to an airspace. Now, the lilac leaf, syringa, is characteristic of a lot of plants on planet Earth, mesophytes. Plants that are adapted to some water, some dry. Later on in this lab, we will be talking about plants that are adapted to either complete submergence or complete access to water, hydrophytes, and xerophytes. So keep our general leaf structure in mind when discussing those variations. Now, of course, we want to also take a look at a corn leaf in cross-section. Corn is a C4 plant, so it will have a very distinct and different structure than our lilac. You'll notice here, as corn is also a monocot, that has many parallel veins, each of these being little vascular bundles, with one central vascular bundle present here. You can see the bundle sheath cells surrounding each of our vascular bundles. You can see the thick epidermis with occasional huge cells, these bulliform cells, being the reason why corn leaves can bend. So they can expand and contract to help promote the bending of the leaf. Now looking at the leaf in a closer cross section, we also have our terms listed. So we obviously know the epidermis. We can identify the mesophyll of the leaf, the filling of the leaf, the conductive tissues, including our giant xylem and our phloem, and our early built xylem. You may see this referred to both in your textbook and in other places online as the protoxylem versus the metaxylem. Proto being the early built stuff and the late built stuff. Small pipes first, big pipes later. We have our bundle sheath where our malate is being shuttled, so that way we can do the dark reaction within this specific cell layer. And then we have bundle sheath extensions, a combination of sclerenchymal, parenchymal, and collenchymal tissue that goes from the bundle sheath to the epidermis on both sides for stability purposes. We also see stomata on the leaves providing air entry, which of course will be very important to the air exchange and providing the plant with the necessary building blocks, carbon dioxide, for sugar production. So again, take a moment here, draw these structures in great detail, and make sure you are labeling things. You will be expected to be able to label slides like this on our midterm. So for our next section, we'll be discussing some of these specific adaptations to different environments. Not that environments needed to be capitalized. Oops. Let's see. Can I change that live? Oh, yeah. Cool. There we go. So we looked at a lot of mesophytes, plants that have the ability to live in moderate water conditions, but we'll also be looking at some of our hydrophytes and our xerophytes. Now, in terms of adaptations specific to our hydrophytes, like Nymphea, the water lily, it's going to need to be a leaf that lives on top of the water. So you will see structural components to the leaf as well as air spaces and the locations of their stomata being inverted. For our xerophytes, or plants adapted to incredibly dry environments, you will see an additional layer of protection against drying out, the hypodermis, as well as stomatal crypts, which means that the stomata are tucked in a little bit further up in the leaf, so that way they can maintain their own levels of humidity in a smaller environment. Think of it being similar to you wearing a scarf in a blizzard. That scarf creates your own little cocoon, so that way you're not constantly breathing in the fresh blizzard air. A ficus leaf, Ficus elastica, rubber tree. You'll notice a very unique feature when looking at this leaf. You'll see a cuticle up top, a large amount of really big cells that have no chloroplasts, and then your palisade mesophyll starting really far down. And then, of course, your vascular bundle, your stomata down on the bottom of the leaf, and the spongy mesophyll. This layer above the palisade mesophyll is the hypodermis, an extra layer underneath the surface of the leaf to help prevent water loss. That is a lot of prevention of water loss. Now, if you're looking at this in class, you may notice that some of the vascular bundles look a little bit weird. Here is a bigger one that's spread out and even has a pith in it. Don't try to focus on drawing these. There's too much going on here. We can talk about it in class if you like. But instead, focus on drawing one of the thinner vascular bundles, a place where the leaf is not so wide, not focused on a main axis. And you can also zoom in at 400x to take a look at the stomatal crypts. You see the fact that this stomata actually has a little bit of a bump to keep it back on the leaf. So the leaf is not quite flat, but instead has a divot inside, again for that scarf-like cocoon. Now next up we have our water lily leaf, Nymphaea. We hopefully will be taking a look at some fresh preparations of Nymphaea leaves underneath our fluorescent microscope. We've gotten some really cool images of this in class, so hopefully stay tuned. But you'll notice that there are airy spaces on the bottom, air sacs, a vascular bundle. Nymphaea species are very unique in their photosynthesis. We won't go into it in this lab, probably a little bit too much. But again, if you want me to, we can talk about the fact that these are an aquatic type of cam plant. And then the other structure that you'll notice that's very different are these really, really big sclerids. These are structural in the leaves. They form different shapes because they are kind of like triangular posts, and you catch them at different locations because they are so absolutely huge. They basically help provide structure to our lily pad that's sitting on top of the water. Top of the lily pad is fairly flat. The bottom has a little bit more curvature to it based on where vascular bundles are. And also, you'll notice a couple of darker regions on the bottom of the epidermis, or the underside epidermis, and that's because you do see some slime producing glands to help lubricate the bottom of the leaf. A little bit closer zoom up does show you that unique bundle sheath structure around the vascular system of Nymphaea. So, having looked at our hydrophyte and our xerophyte, a dry and water adapted plant, two different dry and water adapted plants, our last is a privet leaf, Ligustrum vulgare. And I want y'all to think about what type of environment this plant grows in. Is it more xeric or is it more hydric? Here we have a zoom in on one of these stomata. Bonus points to you if you've found privet anywhere in the area. Last but not least, I wanted y'all to take a look at this leaf abscission zone, a region of programmed cell death in order to allow the leaf to fall off. You see where the cortex, the living cells, are on both sides of this leaf, that they are starting to decay. The blue that was present in there, corresponding to cellular structures internally, has changed its cell wall form to be more lignified, associated with the death of those cells. Once all the cells have died in a stripe, all that's required is a little bit of wind in order to pull this leaf off. But I also think that this leaf abscission zone slide is one of the prettiest slides that we have in our collection, just because of the overwhelming amount of color and in variation in the vascular tissues. So you see a leaf present here, and then adjacent to it you actually see the bud that would have been forming the reproductive structure of the plant. Pretty cool looking, huh?